we will continue to do the review of the examination 3. And I also want to mention the extra credits is right here on the eCampus. If you are never in the lecture, this is your answer. Okay, I wrote very well, very clear. That's your answer. And we also I have all these cameras, all these video recording here, so you can see. We're going to continue to do the review questions. At the end of the review question, I want one of you to volunteer to the front to talk about lactose alcohol. Okay? So let's look at all these questions one by one and give you some of the idea. You don't have to do the first one. This will be re-mentioned again in the final exam regarding FDA, FSIS, and EPA. Okay, here is very simple. A non-protein component of an enzyme that is loosely attached to the protein component is referred as what? Coenzyme, is that right? Okay. If it's non-protein part is loosely attached, this is called coenzyme. How about closely attached? Which one is closely attached? Prostatic group, is that right? And we call this coenzyme. Sometimes we also call it cofactor. Remember? RNA polymerase, you have a sigma factor. There are some more to review. Question number 3. The law of the thermodynamics states that physical and chemical processes occur in such a way that randomness, which means disorder, increases to a maximum. What is this one called? Second, is that right? Second law of thermodynamics. That's easy. The randomness, which means disorder increases. Okay, how are we going to do this question? This is an important question. Cytochroma A, cytochroma C, ferric, and we had oxygen, water, and there is an enzyme, rusty thymine. Okay, here are the voltage. Voltage value would be 0.8, 0.79, 0.77, 0.82, and 0.8. Okay, can you write? These are the electron components in the electron transport chain. I gave you the voltage value. Can you write the flow of the electron? Which one will be on the top first? Which one will be at the bottom? Okay, what is this called? What's the question? What's the knowledge we're going to have to review? This is, we talk about two-half reaction. We say it's more negative, it will be potentially an electron donor. And the more positive, it will be potentially electron acceptor. So, which one is the largest one? Oxygen and the water. Which one is the smallest one? Ferric. Okay, then what are the second one? Cytochroma C. The next one? value is very positive, and this is the lowest one, is 0.77. So relatively, talking about oxygen, this spheric compounds is a possibility is the electron donor. Okay, so the flow away is always like this. That's tested to half reaction. Okay? That's question number 4. So let's look at the other things. We go down. Okay, which of the following processes generates the greatest amount of energy? Fermentation, substrate level phosphorylation, anabolism, glycolysis, anaerobic respiration. Okay, when we look at this question, the first thing we need to take out is anabolism. Because anabolism is combined, is biosynthesis. So they're going to use energy. So C is absolutely is gone. Then you look at these A, B, D, E. Fermentation, substrate level phosphorylation, glycolysis, anaerobic respiration. How many ATP generates through fermentation? 2 ATP, very simple. Substrate level phosphorylation, it really depends. And usually it's 1 ATP sometimes. Okay. Glycolysis, how many ATP generate? 2. If it's an individual cycle. Then which one left? Anaerobic respiration. How many ATP generated between for the anaerobic respiration? The best answer should be between 2 and 38. Depends the electron donor and acceptor, the distance between them. The larger the distance, the more energy going to be generated. Now, when we answer this question, you have to go back to review, which is on examination 3, slide number 2. You have to go back to review around this conclusion table. This is what I said, it is important. That's anaerobic respiration. Okay? 36 or less, what is this conclusion? But I will say 2 to 38, a little bit between them. Because for anaerobic respiration, the terminal electron acceptor is an inorganic molecule other than oxygen. And we mentioned, if the final terminal electron acceptor is oxygen, we call it aerobic respiration. Okay? And what's the detail? It depends. So we didn't give you example, but it depends. So I will say between 2 to 38. Okay, let's look at this. Sulfonamides or antibiotics set function back. How that works? When you look at here, inhibition bacteria grow by binding to, this is wrong, of course. Inhibit enzyme by binding astrocytes, or maybe inhibits synthesis by vitamin by competing the normal structure of the active site of the enzyme involved in the vitamin synthesis. That's a C. Is that right? It's so simple. Sulfur drug. When we mention about the sulfur drug, what we talk about is competitive inhibition. Inhibition of PABA to generate folic acids. Okay. You can go ahead and look at your slides. We'll talk about it. Question number 7. The transfer of electron and hydrogen by a dehydrogenase to an organic molecule as the terminal electron acceptor is called. This is something we mentioned, but I don't think you know. Donor, acceptor. If an electron donor is organic chemical and the electron acceptor is an organic chemical, what is this called? Fermentation. And this is the best definition to describe the fermentation. If the electron donor is an organic chemical, and the electron acceptor is an inorganic chemical, and this inorganic chemical is oxygen, we call it aerobic respiration. Otherwise, if it's others, we call it anaerobic respiration. Okay, so that's a very easy formula there for solve these problems. So when you see the transfer electron of hydrogen by dehydrogenase to an organic molecule as a terminal electron acceptor, it's an organic molecule, so of course you see A, B, C, D, E, F, G even. What is the answer? Is fermentation. It's very simple. Okay, then again, question number 8. Which of the following is the best definition of fermentation? I just said. The best definition is the oxidation of a carbohydrate with organic molecules serving as final electron acceptor. This is the best definition of fermentation. Now, what is that organic molecules? It could be lactic acids, it could be aldehyde, it could be alcohol, so it depends. So question number 8. Question number 9. Which of the following is used as an electron carrier by living organisms? Now, of course, all the choices. NAD+, NADP+, and FAD. This question, you're going to go back to look at your slides. When we talk about this way it is, you're going to have to go back to look at lecture slides. Number 1, when we talk about right here, electron carriers. So nickel-titanium adenine dinucleotides, nickel-titanium adenine dinucleotide, phosphates following adenine dinucleotides. And this is a picture which talks about that and explains the proton motive force. Okay, following this you should study proton motive force and chemosmosis. So that's all the knowledge you have to connect with. Question number 10. When the end product of a pathway inhibits catalysis of the first step of that pathway, this phenomenon is called, very simple, feedback inhibition. Where is the answer? Is that right? Feedback inhibition. And I picked these practice exams is lecture to review all the slides because each question are very corresponded to all these slides. So that's feedback inhibition. You will see the end products. Could it be impact of the intermediate metabolic products, or could it be directly acting the substrate, the beginning of the substrate? Okay, so that's a question number 10. You don't have to question number 11. I already mentioned this one. Okay, we can skip it. You don't have to worry about 13. Let's look at these again. Which of the following can be used as electron acceptor during anaerobic respiration? All of the choices. Where is the answer? I should say other than oxygen. Is that right? So that's why you remember this one. You can do lots of questions. And when you study all these questions, you should combine. That's question number 14. Question 15. Which of the following is not true regarding ATP synthesis? Now, this guy, you're going to have to look back, is right here, when we talk about chemosmosis. Okay. They require proton motive force to make ATP. Yes, that's correct. They spend the inner membrane of mitochondria. We're going to have to see. The proton flow is outwards during ATP synthesis. Is this correct? No. The proton flow should be come back. Because with the proton flow coming back inside of the cell, they trigger the ATP synthesis. So let ADP become ATP. And the very simple, when you look at the slides, you see coming in, and the ATP synthesis. Can you look at here? The slides showed you very clearly. The hypothesis that proton-motive force drives ATP synthesis is called what? Chemosmosis. It's so simple. Question number 17. Organisms that form lactate, ethanol, and carbon dioxide as end products of glucose metabolism are called what? What's the two types of the fermenters? Homo and hetero. Homo is what? What is what we call homo? Fermenter. And hetero. This is generally lactic acid. This is generally others, like alcohol. Okay. Question number 8, you should know very clearly. And be careful. I may not ask you the whole thing. I may ask you the parts. Let's say glycolysis. In the presence of oxygen, how many ATP? In the absence of oxygen, how many ATP? And how many ATP generate during the citric acid cycle? Okay, so you have to be clear about that. And when you wrote this, your calculation needs to be clear. And you don't just write the number there. Okay, you get 1 point when you just write the number there. You have to write the detail. So this is, I don't have to emphasize, it's so important for calculation. Okay, this question. If a yeast metabolizes one molecule of glucose to ethyl alcohol in the absence of oxygen, how many net molecules of ATP would be obtained? It's a choice between A and K. So don't let you guess. Okay, how do you do this one? It's very simple. What is the process of this? Glucose become alcohol. What's the name of this one? Fermentation, is that right? It doesn't matter how complicated the process of fermentation, how many ATP generates? 2. So the answer is C. Okay? Simple. How are aerobic respiration and anaerobic respiration similar? What's the similar of that? Inorganic molecular as an electron acceptor. That's why I said that is the key. When you have this one there, you could have a bunch of questions, maybe 5 questions, you can study them together. So the answer is C. Both utilize inorganic molecular as terminal electron acceptor. Okay, question number 21. Probionic bacteria makes probionic gases, acetic acid, and carbon dioxide in Swiss cheese. When you see those holes, it's a carbon dioxide. These products result from what? It's so simple, fermentation. Okay, where are the questions? What we are talking about? Go back to study slides. This is what we talk about, the application. Okay, I didn't go very detailed. When I teach another class for the chemistry, in the spring semester, we have one single class talk about vinegar, one single class talk about wine and beer, and another class talk about cheese. So we talk about more detail. Here I give you the overall outline. What's the application of the fermentation? And what we talk about is here, probionic acids. Probionic bacteria become carbon dioxide, probionic acid, and the fermentation become of Swiss cheese. Those holes are carbon dioxide. So that's a question. This is easy, I already talked about citric acid. Define metabolic precursor. Name one chemical in this cycle. I already talked about it. So that's the same. In aerobic conditions, it takes sugar to produce the same amount of ATP when compared to the anaerobic conditions. More or less. Should be less, is that right? Yeah, because anaerobic respiration condition is usually the distance between donor and acceptor is small, so it should be the less. Which of the following can be used as electron acceptor during anaerobic respiration? I talked about this already, all of the choices. Central metallic pathway I didn't mention in this semester. Okay, distinguish between first and second thermodynamic law. You should know that. What is the first we're talking about? Energy never generated and created, only transfer between different forms. Potential energy, kinetic energy. What is the second thermodynamic law? Disorder will be increased. Entropy will be increased. Who proposed the chemosmosis theory? Do you know this guy, Peter Mitchell? He's a British scientist. Name one final product generated from pentose phosphate pathway. What is that? Oh, you have multiple choices. Lots of choices you can generate. Which one? NADPH? 5 carbon sugars? Is that right? That's a lot. Precursor of DNA and RNA. That's a lot you can talk. You don't have to worry about that one. We already talked. Okay, so this question we get it done. Then we're going to look for another one. We get done this. We're going to look for the practice number 2. Okay, some of these we haven't gone over. Some of these are basic knowledge. 2 strands of a DNA molecule, or that is they are oriented in opposite directions. Is that easy? What's the terminology? Anti-parallel. Okay. A consent sequence in the untranslated leader sequence of a prokaryote mRNA, which serves as a ribosome binding site, is called what? This is what we talk about. Untranslated leader serve as a ribosome binding site. What is called? Diagonal sequence, is that right? What is this? What they recognize? Is that right? Okay. What is this one for? mRNA. Okay. Question number 3. Aminoacetyl tRNA synthesis categorized covalent addition of what? What to tRNA? Let's look at this. Okay, let's look at the answer. Question should be this one. What's the tRNA they're going to carry on? Which one? Amino acid, is that right? Yeah, that's simple. Oh, we can see. Yeah, that's an amino acids. So genetic information in the DNA is divided into unit cells. It's called what? Genotype, is that right? What is a gene? So DNA. What does a gene mean? So DNA sequence has a genetic information. Okay, it should be called gene. Sorry. The basic difference between RNA and DNA lies in the sugar and the pyrimidine bases. RNA has ribose uracil, whereas DNA has deoxyribose and thymine. Is that correct? Yes. Which of the following is not a pyrimidine? Which one? Is adenine a pyrimidine? Is cytosine a pyrimidine? Is thymine a pyrimidine? Is uracil a pyrimidine? Which one? Is that C? Is that right? Which one is not a pyrimidine? Which one? Then adenine is what? Purine. Okay. Let's go back to very simple. Which of the following is not a complementary base pair usually found in the DNA? Which one? Well, you see A and U, is that right? That's not found here. Which of the following is true about the structure of DNA? Let's see that. Purine pyrimidine base are attached to the one Oh, that's not good. Is that good? Yeah, that's true. That's one prime. Are joined by phosphodiester bonds. Yes, that's correct. Adjacent bases are stacked on each other. The one base pair 0.34 nanometers. So all the choices. That's all. Good. Which one is usually found in RNA but not in DNA? That's easy. Yourself. You. When a DNA molecule is replicated, the daughter molecules often contain one-stranded parental DNA, the one-stranded newly synthesized DNA. This is called semi-conservative. Okay, that's easy. Even in high school, you know that. Question number 11. The Y shape of the DNA molecule where the actual replication process occurs is called what? Replication fork. Okay. During replication, two strands of DNA. Question number 12. Are unwound from one another by enzyme cause what? What is that? When we do unfold, of course, it's not the unwindedness. You never heard about it. Helicases, is that right? Question number 13. The coding sequence in the DNA is normally continuous. That is, it is not interrupted by non-coding sequences. What is that? A, prokaryotes. This is easy. Plants, mammals, and the most insects is eukaryotes. If you choose B, you have to choose C and D. So the A is the only exception. Okay, so that's not a difficult question. The region at the RNA polymerase bound into is the what? Promoter, is that right? We talk about -35, -10. You should know familiar with that. The unexpressed region of the split genes are called what? Remember, we have to cut exon and the combine intron. So what is that? Exon or intron? Introns. Terminal box of E. coli, what is that? Is centered approximately 10 base pair upstream of the starter of the transcription. So it's C. The genetic code is 2B because one codon will specify a particular, more than one codon will specify a particular amino acid. What they call it? Degenerate. So that's B. Which of the following is not a termination code? What's the termination code? UAA, UG, and UAG. AUG is the